Okay, so welcome back to this next video in which we are discussing the LDL receptor. Okay, right, so we're currently in the process of discussing the receptor-mediated endocytosis of the LDL receptor once it has a molecule of LDL bound to it. Okay, so we have shown the LDL molecule binding to the LDL receptor type A repeat domain, and we discussed that what that will enable is it will enable some sort of conformational change in the LDL receptor that allows it to interact with the LDL receptor adapter protein 1. Okay, and the adapter protein is called an adapter protein because really it's going to bind onto the receptor and then it's going to bind onto another protein, and this next protein, which is clathrin, that it's going to bind to is then going to be responsible for the endocytosis. Okay, so really it is just an adapter. It's something that sits in between the LDL receptor and then the protein which mediates endocytosis. Okay, so I don't really have room to discuss what I want to discuss next here, so I'm going to get a new piece of paper now. Okay, right. So before we go any further, I want to discuss with you what clathrin is and what a clathrin triskelion is. Okay, then we'll discuss how this is going to help with endocytosis. Right, okay, so let's discuss what a clathrin triskelion is then. So basically, there are two chains of clathrin. There is a clathrin heavy chain and a clathrin light chain, okay, which are separate proteins. And these are going to assemble into a structure known as a clathrin triskelion. So let me show this. So this is what a clathrin heavy chain looks like. Okay, so let's divide it up into these different domains then. So this little portion over here, and I think I'll colour it in. So this little portion that I'm now about to colour in red, this is known as the N-terminal domain. Okay, so this is the portion near the amino terminus of the clathrin heavy chain, and what we're looking at the moment is the clathrin heavy chain. Okay, so this is the N-terminal domain. Okay, this portion here, which I'll colour in now in green. Okay, so this portion here, this is known as the distal domain, okay, of the clathrin heavy chain. So this is the distal domain. Okay, and this portion here is called the proximal domain. Okay, and I'll colour the proximal domain in, I think, in blue. Okay, now, right at the centre then, you have this final portion here, which I'll colour in turquoise. So this final little portion here, which is known as the trimerization domain. Okay, so in turquoise, this is the trimerization domain. Okay, so this structure that we've just discussed is the clathrin heavy chain. So all of this was just about the clathrin heavy chain. Let me now add on the clathrin light chain onto this picture, and then we'll discuss what a clathrin triskelion is. Okay, so this is the clathrin heavy chain. Basically, the clathrin light chain sits alongside the clathrin heavy chain near this proximal domain here. Okay, so this here, which I'll colour in vivid purple, this is a separate protein which binds to the clathrin heavy chain, and it's called the clathrin light chain. So this is the clathrin light chain. And then the combination of the clathrin heavy chain and the clathrin light chain would then be called a clathrin molecule. Okay, so let's now discuss how we create a clathrin triskelion, which is the main form in which clathrin exists within the cytoplasm. Okay, so basically to create a clathrin triskelion, what you're going to do is you're going to connect three clathrin molecules, which remember consist of a clathrin heavy chain with a clathrin light chain. You're going to connect these three of these together. Okay, so the way you're going to connect them is by connecting the heavy chains at their trimerization domains. So here comes another clathrin heavy chain, like so, and again it will have a clathrin light chain here. And then we'll have another one of these clathrin molecules here. So here's another clathrin heavy chain, and here is its clathrin light chain. Okay, and this structure now is called a clathrin triskelion. Okay, and this is the main form within the cell that clathrin exists in. Okay, so what's now going to happen if we go back to our LDL receptor with LDL bound? Okay, so here we have the plasma membrane. Here is our LDL receptor, this representing the LDL receptor type A repeat domain, okay, in red here. And then we have our LDL molecule bound to this LDL receptor type A repeat domain via the apolipoprotein B100, which I'll colour in turquoise here. Okay, basically we've seen that the binding of the apolipoprotein B100 of the LDL molecule to the LDL receptor type A repeat domain triggers a change in the NPXY domain that makes it visible to this adapter protein within the cytoplasm of the cell. Okay, so here is our NPXY domain, and then what's bound to that is this LDL receptor adapter protein 1, also known as the autosomal recessive hypercholesterolemia protein. Okay, so this is also called the LDL receptor adapter protein 1. So basically what's now going to happen is that clathrin molecules are going to come and attach to this LDL receptor adapter protein 1, and they're going to start polymerizing into a larger structure. Okay, so let me show you this larger structure that they're going to start polymerizing into. So basically they can polymerize into a buckyball structure. Okay, so they can polymerize into a structure that resembles Buckminster fullerene, or, if you like, a football structure, or a soccer ball if you're American. Okay, so basically, let me just try and draw one of these structures out. So basically, Buckminster fullerene is a three-dimensional structure that's made by joining pentagons and hexagons. Okay, so we'll start with a pentagon here. And now, imagine that that pentagon is in the plane. What we're now going to attach to this pentagon is 5 hexagons, okay, one to each of the sides of the pentagon, but they're not going to be within the plane of the piece of paper. Instead, they're going to be coming out of the page towards us. Okay, and they're going to be regular hexagons, so they all have, all the sides are the same length. So here is a hexagon, here is another hexagon, and the reason I've sort of drawn them slightly sort of squashed is because they're supposed to be coming out of the page towards us. Okay, like so. So here's another hexagon here, okay, and then another one here, like so, and then another one here. Okay, so those are all coming out of the page towards us, and then what you're going to do is you're going to attach in these gaps here, you're going to attach more pentagons, okay, and these will be more regular pentagons. Okay, so you'll have another pentagon in here, like so, another pentagon in here, and these are still coming out of the page. They'll be nearly coming vertical now. Okay, another pentagon here, and then another pentagon here, and then you'll attach more hexagons around those, and you can grow the structure around, and it will form a ball, basically. Okay, right, so clathrin triskelions are going to assemble into this sort of a structure. Now let me sort of give you a clue as to how that's going to work. So, basically, if we colour in one of these clathrin triskelions in turquoise, so that's a clathrin heavy chain, and that's a clathrin light chain there, then effectively what we've got is a clathrin heavy chain, let's say there, with a clathrin light chain here. Okay, then we'll have the rest of the triskelion, so let's now draw the rest of the triskelion. So here's another one of these clathrin heavy chains that's within this clathrin triskelion. It will also have a clathrin light chain associated with it. And then here is the final clathrin molecule within the clathrin triskelion there. And again, it will also have a light chain associated with it here. Okay, now the structure doesn't perfectly work. Okay, you will have overlap. You'll take other clathrin triskelions and they'll overlap. with this clathrin triskelion. So for instance, we might center another clathrin triskelion here, and it might have one of its clathrin molecules coming and overlying with this other one. So it won't be perfect. You're not going to get a structure where you only have one portion of clathrin on each of the lines of the Buckminster fullerene. There will be overlap, but basically the clathrin triskelions are going to assemble into one of these buckyballs. So basically what happens is when you bind one of these clathrin molecules to the plasma membrane by binding it to the autosomal recessive hypercholesterolemia protein, or the LDL receptor adapter protein 1. Okay, and I'm now demoting the clathrin triskelion to just being represented as a box. Okay, what's going to then happen is loads of other clathrin molecules are going to join onto that, and they're going to start forming this bucky ball. So what you're going to get is a structure that looks like this, basically. Okay, you are polymerizing the clathrin molecules together like so, okay, to create this bucky ball structure, and basically at least one of these clathrin molecules will be attached to the LDL receptor adapter protein 1, which is then attached to the LDL receptor, which is a transmembrane protein. Okay, so let's show that down here. So it's going to basically pull the membrane inwards as it's forming this, because it's attached down here. Okay, and I'll expand that picture out in a bit more in a moment. Okay, so these blobs are just meant to represent the clathrin molecules assembling into a buckyball, basically. Okay, right, so let's colour in the different portions then. So here is our LDL receptor type A repeat domain. Here is our autosomal recessive hypercholesterolemia protein, so that's in orange here. Okay. We need to put in our LDL molecule, that's very important, so that's here, okay, with the apolipoprotein B100 sitting there, okay, and now we've also got these clathrin molecules attached to the LDL receptor adapter protein 1, which have polymerized into this large structure here now. So they're starting to form a buckyball, basically, which is pinching the membrane off into a vesicle, basically. You're getting this invagination. Okay, and once this is complete, what you will end up with then is a vesicle which will be fully pinched off from the plasma membrane. So you'll have a vesicle like so, with your LDL receptor here. Okay, so here's your LDL receptor. This is your LDL receptor type A repeat domain. Here's your LDL molecule with the apolipoprotein B100 in. So let's colour all of this in. So here is the apolipoprotein B100 in turquoise. Here is the type A repeat domain of the LDL receptor in red. Here is our LDL receptor adapter protein 1, or the autosomal recessive hypercholesterolemia protein, in orange. And then surrounding the entire vesicle now, you will have a coat of clathrin, which will be fully now assembled into this Buckminster fullerene-like structure. Okay, so this is a coat of clathrin proteins which has fully assembled into a Buckminster fullerene structure. So think of a football structure, it's exactly like that. Okay, now I'm not going to attempt to draw that over the vesicle, so I'm just drawing lots of little clathrin proteins all bound together like so. Okay, so these clathrin proteins can be denoted in blue here. Right, so this structure that we now have is called a clathrin-coated endocytic vesicle. Okay, and the first step in the processing of this now is going to be to remove the clathrin coat. Okay, which is a relief to me because it's taken me a long time to draw this clathrin coat. There we go. Right, so this thing that I've got here is a clathrin-coated endocytic vesicle. Now, I want to stress something very important here. This is not an endosome. An endosome is something much, much bigger, and this is where there's a huge amount of misinformation out there. People simplify these pathways to oversimplified versions, okay? So I'm going to give you the better version where we don't oversimplify things, and this is not an endosome. Okay, an endosome is an intracellular organelle, which we'll come to in a moment. This is an endocytic vesicle. So this is a clathrin-coated endocytic vesicle. Okay, and the first thing that's going to happen to this clathrin-coated endocytic vesicle is that it's going to uncoat. So the clathrin is going to come off the vesicle, basically, and what you're going to end up with is the vesicle membrane here, like so. Then we've got our LDL receptor here, okay, and then we've got our LDL molecule within the endocytic vesicle here. And of course, the LDL receptor type A repeat domain will still be bound to the apolipoprotein B100 at this point. Okay, like so. So here is our apolipoprotein B100. Here is our LDL receptor type A repeat domain. And this structure that we've now got is just an endocytic vesicle. Okay, and this is now going to go to an endosome. Okay, so this process of taking the clathrin off, this was just called uncoating. Right, so let me now discuss with you what an endosome actually is. So an endosome is another intracellular organelle, and they're involved in the processing of endocytic vesicles which are being pinched off from the membrane. Okay, and by the way, this entire process of endocytosing the LDL receptor with its cargo bound to it is known as receptor-mediated endocytosis. So this is an endocytic process, and because the receptor was totally in charge here and caused the whole endocytosis of the LDL molecule, it's called receptor-mediated endocytosis. Right, and the LDL receptor is one of the archetypal examples of receptor-mediated endocytosis. Okay, right, so what's now going to happen is this endocytic vesicle is going to go and join with one of these much bigger intracellular organelles known as an endosome. Okay, right, so let me just draw you a little picture of a cell to compare the difference in size between an endocytic vesicle and the endosome. Okay, so if this is our cell, here's our nucleus. Basically, the first endosome that you are going to go to is something called the early endosome. This is an intracellular organelle. Okay, so this is called the early endosome. It's a membrane-bound intracellular organelle. Okay, where to contrast the size difference between the early endosome and an endocytic vesicle, that's like your endocytic vesicle here. It's coming from the plasma membrane, and it's now going to fuse with this much bigger structure called the early endosome. So, here is our early endosome here, like so. And basically what's going to happen is that this endocytic vesicle is going to come over here and it's going to fuse with the membrane of the early endosome. And what's going to happen is, if you can imagine, this is our endocytic vesicle here, fusing with our early endosome. Here's the LDL receptor. Here is our LDL molecule here. So again, I'll colour in the LDL receptor type A repeat domain in red, and I'll mark the apolipoprotein B100 in turquoise there. And basically imagine that that line isn't there. The endocytic vesicle is fused with the early endosome, as this is, and temporarily the LDL receptor with its cargo loaded on will be within the early endosome. Okay, so here is the LDL receptor with its LDL bound to it. Okay, so here's the apolipoprotein B100 in turquoise, and here's the LDL receptor type A repeat domain. And now what's going to happen is it's not going to stay within the early endosome for long. What's going to happen is you're going to bud a vesicle back off from the early endosome, okay, and it's going to go to another endosome now, another endosome called the late endosome. Okay, so here is our LDL receptor with the type A LDL repeat domain. Okay, and here is our LDL with the apolipoprotein B100, and here is our LDL receptor type A repeat domain. Right, and this vesicle, which has budded off the early endosome, is now going to go to another endosome, which is called the late endosome, and it's the same principle. The late endosome is a large membrane-bound organelle, basically. So this now here, this is the late endosome. And again, what's going to happen is this little vesicle that's transported the LDL receptor with its cargo from the early endosome to the late endosome is going to fuse with the late endosome, and the LDL receptor, along with its cargo, will end up within the late endosome. Now, something interesting is now going to happen here. Okay? Basically, in the late endosome, you have a very, very acidic environment. Okay, so there is a low pH within the late endosome. It's very, very acidic, high proton concentration in here. And basically, under that high proton concentration, what's going to happen is the LDL receptor is going to release the LDL molecule. Okay, so let's go over the page and continue this discussion. So, here we have our late endosome here, and basically we'll start with the LDL receptor being delivered with its cargo attached to it here. Whoops. Okay, so here is the LDL receptor type A repeat domain, and here is the apolipoprotein B100 in turquoise. Right, however, because of the extremely acidic pH within the late endosome here, i.e. it's very, very high proton concentration, low pH, okay, what's going to happen is the LDL receptor and the LDL are going to split apart, okay, like so. So here we have the LDL with its apolipoprotein B100, and then we also have the LDL receptor type A repeat domain. Okay, so remember the interaction between the LDL molecule and the LDL receptor type A repeat domain was governed by these acidic residues which were in the LDL receptor and the basic residues which were in the apolipoprotein B100. Okay, you needed the acidic residues to have lost their proton, and you needed the basic residues to have gained a proton. If you have a too acidic environment, these acid molecules in the LDL receptor type A repeat domain are going to get reprotonated, basically, and they're going to become neutral, which means they're not going to be able to interact with the positive charge of the apolipoprotein B100 anymore. Okay, so the interaction will fall apart because of that extremely high proton concentration. Now, what's then going to happen is you're going to bud off little vesicles off this late endosome, which will take the LDL receptor out of the late endosome. Okay, so you're going to rescue the LDL receptor, basically. Okay, so now that they've fallen apart, you can take the LDL receptor without taking the LDL. So little vesicles are going to bud off, and these are going to take the LDL receptor back to the plasma membrane. Okay, so the LDL receptor will be recycled to the plasma membrane. So this is the recycling of the LDL receptor. Okay. Right, meanwhile, it's not so good news for the LDL molecule. The LDL molecule is also going to go into a vesicle. Okay, so you're going to pinch off a vesicle, like so, which will contain our LDL molecule, okay, here. And this is not going to go back to the plasma membrane. This is going to go to a membrane-bound structure known as a lysosome. Okay, so it's going to go on to a structure called a lysosome here. So this is our lysosome, and basically the lysosome will be full of molecules, enzymes, which can break down the contents of the LDL molecule. So what's going to happen is this little vesicle that from the late endosome is going to go and fuse with the lysosome. Okay, so this is the late endosome. It will give off a little vesicle which will contain the LDL molecule, and this little vesicle containing the LDL molecule will go and fuse with a lysosome which contains all sorts of enzymes which can break down the LDL molecule. Okay, So, specifically, what's going to happen here? Remember what's present within our LDL molecule. We have the apolipoprotein B100, the phospholipids, okay, so mainly lecithin, and then we have cholesterol, and we have this core of cholesterol esters. One of the key transformations that is going to occur is that these cholesterol esters are going to be broken down by enzymes within the lysosome to free cholesterol and free fatty acids. So the key thing that this is going to deliver into the cell is cholesterol. Okay, right. And we'll continue this discussion in the next video when we'll see which tissues have the LDL receptor on their surface.